Hi, everybody.
per
I'm going to go
volume
ahead and go over your free
cytoplasm.
response from
All
Friday.
right,
So the first
this
one is
when you look
identify
at
the
this,
rectangular cell that
the small
represents
surface area to volume is rectangular solid 1. So that is going to be the answer. So that's going to be rectangular solid 1 as the smallest surface area to volume ratio. So therefore, it's going to have the smallest amount of plasma membrane per volume of cytoplasm. Right, that's saying the same thing. So that is the answer to that one. All right, letter B, describe the relationship between the dimensions of the rectangular solids and their surface area to volume ratios. All right, so if I look at this, we have length, height, width. All right, here what's happening is the length is getting larger. Here the height, as we go down, okay, the height is getting, this is getting higher. The length, or I guess it's getting longer. Okay, this is getting smaller. The width is increasing. So I basically am going to change that for clarity for you guys. I swear. So I think you got the idea, but I'm just going to write these clearly for you guys. So the length is increasing. The height is decreasing. And then the width is increasing when I look at these. All right, so hopefully, this is you're not gonna be talking out loud like I am, or even to yourself this long. All right, so the relationship between the dimensions, which those are the dimensions, and then the surface area to volume ratio is over here. Okay, that's increasing. So you just need to do exactly what the question says. All right, so describe the relationship. So I would say, as the rectangle increases the length, and if you're thinking of a rectangle, you could say as it gets longer, decreases the height. And if you're thinking about it, it's getting thinner, right? Decreases the height and increases with the surface area to volume ratio. Get a little. I'm not able to use my eraser. Okay, so you know that doesn't say ration. The ratio, that is going to increase. All right, so that is the answer to B. All right, let's see what C says. Using data from the model, evaluate the hypothesis that cells Using data from the model, evaluate the hypothesis that cells shaped like rectangular cells 1 and 2 are better suited for slow metabolism and the long-term storage of energy-containing molecules than are cells shaped like rectangular solids 4 and 5. All right, so 1 and 2, I believe, had a smaller surface area to volume ratio, and 4 and 5 had a larger cell surface area to volume ratio. So I'm going to go back and look. We're comparing 1 and 2. So here's 1 and 2. They're 5 and 6, and 4 and 5, they have a larger surface area to volume ratio. So this small surface area to volume ratio, that means that cells cannot exchange things efficiently. So that makes sense that then they would be able to store things longer and also have a slower metabolism. So I am going to go ahead and write that solids 1 and 2 have a smaller than solids 4 and 5. This would allow them to store energy molecules longer than ones with a larger surface area to volume ratio. Okay, so it does say are better suited for slow metabolism and the long-term storage of energy-containing molecules in our cells shaped like rectangular cells, 4 and 5. So I noticed that I didn't put the slow metabolism. So I would want to go ahead and add that in. That's something I missed. This would allow for them to store energy molecules longer than ones with a larger surface area to volume ratio. This also would provide for slower metabolism. All right, and we'll check this against the rubric to see what it says. All right, so I ran into the next question right here, D. Based on the trends shown in the data, explain why some rectangular cells include folds or projections within their plasma membrane. All right, so a lot of villi is in there, and so this is going to I would start off by having folds, or does it say projections? Or projections increases the surface area. All right, and so you have to think like, all right, that increases the surface area. All right, and that's going to allow for better, that's going to allow for a better exchange of materials. That's going to allow for the surface area to a volume ratio. That's going to increase it without decreasing the cell size or changing the shape of the cell. So I'm going to say without changing the size of the cell. Okay, so let's take a look. All right, rectangular solid 1, the response identifies rectangular solid 1 with the smallest surface area to volume ratio as a cell with smallest amount of plasma membrane. Okay, so that is 1 point. Select point value for scoring, so the next one is 1. Did I miss it? B. B. The response describes that the rectangle becomes longer, wider, and thinner. The surface area to volume ratio increases. All right, so this was said differently. Remember, I went length, did this. The width, as the length increases, the width increases, and then thinner, that would have been the height. The surface area to volume ratio increases. All right, so that's what we said. So that is going to be another point. So that's 2 points. Rectangular solid 1 and 2 are better for storage. All right, so we said that. The explanation is that the rectangular solid identifies better for storage because of lower surface area to volume ratios. We said that. I did go into the metabolism, but it doesn't seem like that's needed. I don't know why, because it asked that. So I do want you guys to do whatever it asks. All right, the response indicates that folded projections produce cells with increased surface area to volume ratios without reducing the cell size. So like I did not say ratio, but did it ask that? So I said surface area without reducing the cell size or changing the shape. So this one, let's look back at this question. D. Some rectangular cells include projections in their plasma membrane. Why some do? I think that's a point. I only said surface area, but it does increase the surface area without changing the size of the cell. So it does not increase surface area to volume ratios without reducing the cell size or changing the basic cell shape. You know what, this one I'm not sure. Like I feel I have it, but what we would do is on the error of caution say, you're not going to get that. So this is kind of realistic. Like, let's see if you guys made sure, like, I think I made a mistake there not putting in the ratio. But basically that's what the folds do, is increase the surface area. So most likely I feel if this was at a read that this would get a point. But based on this rubric, it's a little weak based on this rubric, but who knows? Because I did say without changing its size. I didn't bring in the shape. I don't know. I don't know about that one. I'm going to say that I would get that, but that's not for sure. All right, let's look at this question. All right, glucose and sodium move from the lumen of the small intestine. All right, here's the lumen. of the small intestine into the blood via transport proteins and epithelial cell linings in the small intestines. Right, here's the epithelial lining in the intestine. Here's the blood. Based on figure 1, describe the direct source of energy used to move glucose into the epithelial cell from the intestinal lumen. So we want to find lumen, and we're going into the epithelial cell. So glucose, how is it getting in is what that's happening. Like, describe what's going on. All right, so basically it looks like it's going in with the energy of this sodium. And this says there's a sodium glucose symporter, so this is through co-transport. All right, but basically if I go back again, it says describe the direct source of energy used to move. So they want to know what is the energy that's moving it. All right, so I would say it's the energy of those protons. So which protons? So I would say for the description, I would say sodium basically the sodium gradient. And the reason why I want to say gradient, I should have pointed this out, but I can see that why would sodium want to move in? Because look over here at the sodium potassium pump. Sodium is moving out. So remember, these ions will move from high to low concentration. So if this is coming out, this coming out is going to pull this sodium in. So the energy of this sodium is going to bring the glucose in, which is co-transport. So the sodium gradient is the source of energy. And then now it says, explain how this system maximizes glucose absorption from the intestinal lumen. So we're here again into the epithelial cell and from the epithelial cell into the blood. So basically it wants you to explain how this all works. All right, how does the glucose go from the inside of the intestines into the epithelial layer to the blood? All right, super important because we know that we need glucose for cellular respiration to get energy. So remember, like you digest your food, most of it's in the intestine. All right, so basically I would say glucose enters the epithelial cells by co-transport of sodium regulated by the sodium potassium pump. Okay, so again over here, sodium comes out from the pump, potassium in, but then sodium is brought in. Okay, and so that's how glucose gets in there. Now, to me, how does glucose get into the blood? Glucose is going through this transport protein right here. And even told you that there's protein. So you know that that GLUT2 transporter is a protein, but you should probably be able to infer that. So basically, what is it called when glucose or a molecule is going to move from high concentration to low concentration? No energy is required, but there's a protein. So that's going to be facilitated diffusion. So I would say glucose moves from, you know what, from the epithelial cells and is absorbed by the blood by facilitated diffusion. All right, that's what I would do. I'm going to go back. So do I hit epithelial cells and epithelial lumen to epithelial cells and epithelial cells to the blood? Yes, I did. So sodium gradient is a source of energy. So sodium gradient is a source of energy. Glucose enters epithelial cells by co-transport of sodium regulated by the sodium-potassium pump. Glucose moves from the epithelial cells and absorbed by the blood by facilitated diffusion. I threw the word absorb in there because I saw it was up here and I realized I wasn't using it. All right, let's check it out. All right, energy is the sodium gradient. Let's look at this explanation. 2 points. The sodium-potassium pump maintains the sodium concentration gradient, allows for the co-transport of glucose. All right, didn't say it exactly like that, but we got that. The symport inflow of glucose maintains a glucose concentration gradient between the epithelial cells and the blood and allows for facilitated diffusion of glucose. All right, I think that we got those 2 points. Here's something else. The microvilli folds lumen side of the epithelial cell to provide more surface area for uptake of, and I'm sure it says glucose into the epithelial cell. Right, so that even talks about surface area again. So yeah, I would say that we got all 3 points for this. All right, one thing I want to point out about this test, what my personal feeling is that you have to know these processes. All right, so I feel there will be a disruption. All right, so seeing if you can understand different processes, I think you're going to be presented with some type of disruption, so that will test your knowledge on whether or not you understand the material. Okay, so that was it. So that's 1, 2, 3, 4. There is a total of 7 points for this whole thing. So it's a little shy. I think that there will be, you know, at least a 10 point is what we should try. So I think we can try to answer a couple more things.